Hello, I'm Guido Di Gallo, a senior toxicologist in Accelera, responsible for ATMP programs. In this presentation, I will explain you the experience acquired by Accelera in the management and development of ATMP projects, in particular CAR and stem cells. In this presentation, I will go through the regulatory background, then move to a description of CAR-T programs that were conducted in Accelera, and in the end, in a few minutes, I will go also through a stem cell program that we have done. ATMP programs have to follow specific guidelines. In this page, you can see the links to the European and American agency site where the guidelines are present. They mainly focus on efficacy, quality, safety of cell therapy and tissue engineering compounds, and of course of gene therapy compounds. In addition to the specific guidelines, also the standard guidelines like the ICH guidelines for biotechnology-derived products have to be taken into account. In Europe, you can contact the European agency to check if your compound can be classified as ATMP. Main points that have to be taken into consideration in non-clinical studies for ATMPs, and in this case are mainly related to genetically modified somatic cells or to stem cells, are the biodistribution, migration, and persistence of the cells or of the gene, release of transfer vector in vivo, induced cellular changes, the in vivo behavior and activity for the transduced cells, any unwanted immune response, and the encapsulated cell status. We have to evaluate the in vivo behavior and activity of transduced cells, the appropriate distribution, trafficking and localization and persistence of the genetically modified cells, the expression, activity, localization and persistence of the relevant gene, the therapeutic effects of the transduced cells or of the transduced gene. In addition, we have to evaluate any unwanted immune response that could be related from the host to the graft or from the graft to the host. Guidelines require that the animal species model have to be carefully selected, it must be sensitive, could be transgenic or immunosuppressed, and then a single dose could be accepted. Of course, the immunogenicity and the immunotoxicity of the compound has also to be evaluated. For example, for DNA vaccines, the expected response has to be controlled, and immune responses or autoimmune reaction have to be evaluated. Another relevant point, the study duration and the dosing frequency. Single-dose study evaluating safety endpoints can be replaced by data derived from the efficacy study if available. Repeated dose studies are necessary if repeated doses are foreseen in the clinic. Pivotal studies for the clinical submission must be GLP regulated. The authorities recognize that since the novelty approach to these studies and to this compound, not all activities can be conducted according to GLP, but any deviation have to be justified. In summary, main concerns of regulators regards the distribution and fate of the cells, the safety and the unwanted effect of the ATMPs, and the tumorigenicity capability of the ATMPs. Keeping in mind regulatory requirements, we have to approach each ATMP project with a case-by-case attitude. We have to evaluate the CAR-T efficacy and safety. The CAR, while doing so, we have to take into consideration the delivery approach of the CAR gene into the cell, the in vitro culture condition, the constituent of the CAR construct, the type of host T cells, and their possible effects on the in vivo animals. The preclinical in vivo assessment of the efficacy and safety is essential for the dosage determination and for risk management of this type of therapy. We have also to take into consideration that many CAR T-cell programs are actually in the clinic, some are also approved, and what are the side effects or adverse events that were reported for such programs. There were cytokine release syndromes, B-cell aplasia, neurotoxicity, including seizure-like activity, and cerebral edema. These last adverse events were acute and manageable in the clinic. Cell product aspects that have to be taken into consideration are the genetic relationship of the cells to the patient. They can be autologous or allogeneic therapies, and in some cases even xenogenic. Then we have to consider if they are mature cells or stem cells products, and at the end also the expected effect of the cell product. Other relevant points are the source of the cells, the extent of the ex vivo manipulation format, the selection, purification, or expansion, and even the activation of the cells, the vector used, the proliferation and integration of survival of this vector, and based on this characteristic, also what can be the control arm to be selected for the studies. Based on the characteristic of the compound, we have to evaluate the fate of the cells after administration, engraftment, migration, differentiation, persistency of the cells. Then we have to consider that the duration of the observation of these studies is usually longer than standard single-dose studies, since cells are deemed to persist and to induce long-term effect. Also, the tumorigenicity of these cells have to be evaluated. Also, the proliferation in an unwanted quantity or in an unwanted location of these cells have to be taken into consideration. Then we have to consider immunologic reaction, the probability of host immune response to the administered cells, and the probability of cells versus host immune response, graft versus host disease. Administration site reaction, and the potential inflammatory response in target or non-target tissues. Since cell therapy products are based on human cells, standard immunocompetent animals are not suitable for pharmacological and toxicological study due to the xenogenic immune reaction. In some cases, homologous compound can be used, but most commonly immune deficient animals are used for these projects. Based on literature and on our experience, immunodeficient animals can address the pharmacodynamics and safety aspects, even if not all of them. There is a large and growing number of published data from on-CAR T-cell products that are performed in NSG mice. In Acevera, we supported different projects with CAR T-cells and stem cells. We had two projects intended for autologous CAR T-cell therapy and 4 projects with allogeneic CAR T-cell therapy. We conducted efficacy studies, anti-tumoral, and safety and tolerability studies, including preliminary studies and GLP studies. Stem cell project included homologous stem cells in a canine model of Duchenne muscular dystrophy, and autologous stem cells in an NHP model, and then human stem cells in immunodeficient mice. CAR T-cells program were conducted in immunodeficient NSG mice. We conducted efficacy studies, anti-tumoral studies, pilot studies for cell engraftment and tolerability, toxicology studies with biodistribution and persistency. We had full pathology with macro and microscopic examination, and the biodistribution was investigated by flow cytometry analysis, immunohistochemistry, and PCR. As regards the anti-tumor efficacy studies, most of the studies were conducted on three tumors model, but the selection of the model depend on the target of the CAR gene. The endpoints were the survival, the in vivo luminescence evaluation of the tumor growth, and the presence of CAR T and tumor cells in blood, spleen, bone marrow at different time points. Toxicity and biodistribution studies were conducted as a single or repeated dose administration in NSG mice, usually by intravenous injection. The study duration was 3 to 6 months for the GLP studies, with sacrifice at part of the mice at an intermediate endpoint. During the study, clinical pathology evaluation was conducted. A full tissue list was examined according to the guidelines, and the cell distribution in selected tissue was also evaluated. While starting the cell therapy program, preliminary tests and in consideration have to be taken into account. The test item method of use have to be considered. We need to take into account the storage condition of the test item, how the test item is provided to etc., the type of formulation required, the method of preparation, the frequency of preparation, taking into account the time required for dosing and the survival of the cells. We have to consider the dose concentration, taking into account volume limitation for dosing, the storage condition after the preparation, and then the test formulation checks and stability and the viability of the cells. Cell thawing and cell preparation protocols require an ad hoc approach. Different thawing protocols are in place, but they have to be adapted to sponsor and material requests. All the procedures have to be recorded, and if requested, the cell suspension can be cultured for a short time before administration. Cell suspensions have to be checked for concentration, cell count, and viability. We use a Barker chamber, and for viability is controlled using a Malassez method. In addition, this check can be performed at different time points, so we have a stability and instruction for use of the cell suspension. Additional checks can be conducted using the flow cytometry and PCR. Bioanalytical methods have to be in place before starting pivotal studies. The method implied have to be specific. They will be able to recognize human cells versus CAR T cells. They must be sensitive. We have to understand in advance the tissue selection on which to perform the analysis. PCR method can be used, either quantitative or digital droplet PCR. Immunohistochemistry can be applied and different markers can be used. Flow cytometry marker selection have to be discussed on a case by case. A standard list of the project, a CAR-T project related activity is presented in this slide. As I told you, we have formulation activity that goes from a method set up for the preparation to concentration check, viability or stability data. We have method for bioanalytical assays like PCR, immunohistochemistry, and flow cytometry. Then we have the in vivo study with efficacy, a DRF study, and GLP pivotal studies. In the next section of the presentation, I will give you some example of studies performed for CAR T cells program. Conventional pharmacokinetic studies are not useful for these compounds. The evaluation of the cell migration and persistence is conducted, if possible, by evaluating viability, distribution, growth, and differentiation of cells. Cells are usually administered by EV in NSG mice. The administration can be single or repeated depending on the compound characteristic. A control arm is usually present in these studies, and the animals are given unmodified T cells or mock modified T cells. In this slide, a classical design of a MTD or DRF study is presented. We usually have only two dose levels, since the high dose is usually limited by the suspension stability and the possibility to administer the cells to animals. Control arm is useful to check difference in the reactivity of the cells against the host. The observation period of 8 weeks is relevant since it allows to start the reaction of the cells against the mice. Flow cytometry is conducted on circulating blood, usually at early time point, to check for cell presence in the peripheral circulation. Histology is usually performed at the end of the observation period on selected tissues. After that, the main pivotal GLP study has to be conducted. The study doses are selected on the basis of the preliminary study, and the duration of the study is usually prolonged to allow for the emergence of any delayed effect. An interim sacrifice is in any case included in this study to evaluate the fate of the cells at early time points. Different examinations are planned during the study. The number of animals per group is usually reduced in comparison to standard toxicological studies, but this in the past has been accepted by regulatory authorities. The fate of the cells in these studies, the cell fate in non-tumor bearing mice can be different. CAR T cells without their target can reach a dormant status, but this depends also on the possible activation of human CAR T cells against the mice tissue. In some cases where the human cells are rendered host tolerant, they can reach dormant status and be hidden and sequestered in some tissues like the bone marrow or the spleen. In this case, they are difficult to find and to identify after a long-term observation period. If cells are activated against the mouse, they can proliferate and be aggressive against the host tissues, giving the graft versus host disease. And in this case, their effect is and number is much easier to be found in mice. During the studies, overall survival, clinical observation, and body weight are recorded. Clinical pathology is performed at different time points. Local tolerance of the injection site is controlled. Macro and microscopic examination are performed at the end of the study and at scheduled and unscheduled sacrifice. Cytokines are also measured usually during early time points for any early reaction. Flow cytometry to detect the cells generally in peripheral blood is performed at early time points, but can be also used on a few tissues at sacrifice to check for the proliferation, phenotype, and persistence of T cells. GVHD signs are checked over the course of the experiment, and diagnosis of GVHD can be also included in the pathology examination. Immunohistochemistry to detect the cells in major organs can be performed, and PCR and expression of selected genes is performed at selected time points in different tissues and organs. In this slide, you can observe the different measurements performed during a study when CAR T cells were designed for allogenic administration. They were less reactive towards the host. Unmodified T cells reacted against the host, giving signs of graft versus host disease, and that was dose dependent and time dependent. The same observation of dose and time dependency can be observed in cytokines. Flow cytometry is able to identify the CAR T cells in comparison with normal human unmodified T cells. At histopathological examination, different signs of graft versus host findings are present in different organs. In this example, you can see the lungs, normal appearance on the left, and pathology findings on the right. Biodistribution and persistence of cells have to be evaluated in these studies. PCR is a very useful tool and can identify human cells in the mouse tissues and CAR T cells in mouse tissues at different time points. In this case, we have an example where primers for mouse, human, and CAR gene were evaluated after 2 and 13 weeks of study. The animals given the unmodified cells, the first two columns, show that there are no CAR T primer, while the human cells are present in the tissue listed. Other tissues were examined but not reported in this example. CAR T cells treated mouse show that human cells are present as well, and CAR T cells presence is very, very similar as a percentage over the mouse tissue, meaning that all human cells present in these tissues are CAR T cells. In the next few slides, I will show you another example on stem cells project. Duchenne muscular dystrophy is an untreatable genetic disease that affects primarily the skeletal muscle, causing fiber degeneration and progressive paralysis and death. There is no therapeutic strategies at the moment, and promising gene and cell therapy are limited by high cost. Three therapeutic approaches are possible: drug therapy, like with utropine, that seems to slow down the muscular disease; gene therapy, where the mutated gene can be replaced; or cell therapy with stem cells. The model is the golden retriever muscular disease that show a dystrophin protein defect similar to the human condition. Since the dog disease model was available, an homologous mesangioblast therapy was scheduled and performed to allow the investigation of efficacy and safety for these stem cells. The study of efficacy was performed in France with golden retriever spontaneous pathology at a university level. Safety evaluation was conducted in our laboratory with well-established and controlled preparation of canine mesoangioblast. Then the study was conducted by repeated administration at different dose levels in young beagle dogs. The animals underwent chemical immunosuppression to avoid the rejection and also to mimic the clinical application. Standard GLP repeated study procedure were applied, and based on these results, a clinical study was allowed to start in Europe by administration of a donor HLA-matched allogenic mesangioblasts in young children. Thank you for your attention.